All right.
Nothing like a curveball.
All right. Don't throw things at Brian. We need him up there.
Take a little Dutch.
All right. So in the midst of the chaos here, it seems to be working. So what we're going to do today is march into, again, continuing our march into the tree of life itself. And we're going to spend most of today talking about various aspects of the three domains. So we sort of led into this in the previous lecture, talking about the tree of life a little bit. I'm going to review that a tiny bit, and then talk about some aspects of the three domains. And in the next lecture, we're going to focus in on just a portion of the tree of life with some stuff on bacteria and archaea. So one of the things that we're going to do today, and in the next couple of lectures maybe, is refer a lot to features of bacteria, archaea, or eukaryotes. Some of their cellular features, some of their molecular details of how, say, transcription and translation and other processes work in some of these organisms. And we're going to assume you have learned some of this and will recall some of this. And if you don't remember any of these sort of features of a eukaryotic cell or how some of the basic features of, say, transcription and translation work and other molecular processes, you should look at, in total, this is about maybe 15 pages. There's a tiny bit in section 1.1. There's a tiny bit in section 1.2 about the different sort of main lineages of organisms. There's a little bit in section 4.4 on, again, the main features of different organisms. And then this part you probably should read and familiarize yourselves with, basically about page 77 to 84 or so, as well as like 84 to 87, I think, or 88. That's the general features of a prokaryotic cell. Those are the organisms that do not have their DNA packaged in the nucleus, and the general features of a eukaryotic cell. And we're going to go over many of these in the next 2 days, but you should also read up on this. As I said before, there's going to be a bonus question on the test that relates to some of the Nobel Prizes. Fortunately for me, writing the bonus question, today's Nobel Prize that was awarded in physiology and medicine is very relevant to the class and to my section of the class because it was awarded for how organisms like us recognize microbes, parts of the what's called innate immune system and parts of the adaptive immune system. You should read up on little detail of the Nobel Prize and the biology. I might ask a question related to that. Again, I'm not going to tell you exactly what it's going to be because it's a bonus point question. So now what I'm going to do is, again, take you through this a little bit of stuff we already covered, and I'm just going to reiterate it and talk about it in a slightly different way about the three domains, the tree of life being divided up into these three main lineages. So again, this was predominantly done by one person, and subsequently his analysis has held up with a lot of different data and a lot of different analyses since then. And this was done by this guy, Carl Woese, who was at, and still is at, University of Illinois when he did this. And what he basically did was he said, okay, well, all of these previous trees of life that tried to shove all the organisms on the planet together did a really poor job with microbes. They mostly just smushed microbes into the bottom of the tree or guessed where microbes might be. And the main reason for this was that when people were trying to build a tree of life, say the one I showed you from Haeckel or Whitaker or other people, they didn't have features that they could readily compare. That is, characters that they could readily compare between eukaryotes, and in particular multicellular eukaryotes, and microbes, including the single-celled eukaryotes as well as the bacteria, or what they called then the prokaryotes, the organisms without nuclei. And so in general, any attempt to sort of include them on one tree was mostly guessing in a way. And what Woese did was said, okay, we should be able to do this. There are universal genes. There are these genes that make up the ribosome that we talked about last time, components of the ribosome, different proteins in the ribosome, and then some RNA molecules are part of the ribosome. So the way it works in the central dogma, DNA is transcribed into RNA, is translated into proteins. There actually are some RNAs that are never translated into proteins. tRNA, which is used to read the genetic code, is one of them. Ribosomal RNA is another. And so inside this machine, the ribosome that all organisms have, are components that are similar across all organisms. And that includes some of these proteins in the ribosomal RNA. And what Woese realized was that even though everybody has those components, and therefore we can't build a tree out of presence of those components, because that's plus, 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 plus across all organisms, you can look at the sequence of the DNA in the genome of these organisms that encodes these different components. And that sequence of the DNA is different between different organisms. So for the small subunit ribosomal RNA in organisms, humans have a small subunit ribosomal RNA gene, chickens have a small subunit ribosomal RNA gene, bacteria, archaea, all these other organisms have these small subunit ribosomal RNA genes. They're all similar to each other but not identical. And you can treat this gene that's about 1,500 letters long as 1,500 different characters in each organism. And you can line up the sequence from organism 1 to the sequence from organism 2 to the sequence from organism 3, and then have 1,500 different characters at which there is some variation in the exact base used by different organisms. And you can build a tree from that. So that's what he did. He took advantage of this feature. If you look at the RNA molecules in the ribosome, they fold over. They're going to play with that for a second, so I'll just talk while they do it. They fold over, these ribosomal RNAs fold over into these what are called secondary structures, where the base pairing rules, just like between two strands of DNA where C lines up with G, the C can line up with a G in the ribosomal RNA, and it forms this structure. That structure is important to its function. You can line up the ribosomal RNAs from different organisms by comparing their structure. So one organism might have C here and G there. Another organism might have A here and T there, but you can still line them up because they have the same general structure. So you now line these organisms up and compare them, and this sort of outlines the logic behind that. So all cellular organisms have ribosomes. They all have these homologous subunits. What Woese did was he read the string of letters inside the genes for the ribosomal RNAs in these different organisms. And again, they're very similar to each other, but not identical. And each position can be considered a distinct character trait. And he actually looked at the RNA. So RNA uses A, C, U, and G as the four bases. DNA uses A, C, T, and G, but that subtle detail doesn't really matter for our purposes here. There are four different character states for each character in the ribosomal RNA. So what you can make is what's called an alignment. This alignment, each row in this alignment is a different organism. This is just focus in on the top here. Organism 1, organism 2, organism 3, etc. This is a data matrix. It's conceptually identical to the data matrix we used to build evolutionary trees before. Each row is a taxon. Each column is a character, a different character in the ribosomal RNA. And they vary in the exact base that is used in the different characters. And again, this is a great thing because within one gene you can have thousands of individual characters to build your evolutionary tree with. And that's what Woese basically did. So this is analogous to lining up the bones of different vertebrates with each other. So if you line up the femur bones and the radius bones and the hip bones, etc., you compare the different bones to each other. Those are each your characters. Each position in the ribosomal RNA is sort of the equivalent of a different bone. And this is called an alignment because you're lining up the sequences from different organisms. And again, you can use this structure to make sure your alignment is pretty good, just like if you were comparing two different vertebrates and one of them had an extra bone, say in between the humerus and the radius and ulna, somewhere along here, you wouldn't line up that bone with bones in the head of another organism. And that's what this structure of the ribosomal RNA allows you to do while they play with our... So Woese did this, and he read the sequences from lots of different organisms. And he lined them up to each other, and he compared them. And I mentioned this before. We're not going to go into the detail. It turns out he used, in his original analyses, the third class of phylogenetic methods that we haven't talked a lot about, called distance methods. It turns out it doesn't matter which method you use. You get the same pattern. But from a historical point of view, it's interesting that he used these distance-based methods. And this is what he found. Instead of there being two groups of organisms on the planet, which is what sort of most people thought at the time, where they said there were eukaryotes, that is, organisms where the DNA is packaged in a nucleus, and prokaryotes, that is, organisms where the DNA is not packaged in a nucleus. Woese found that there were three groups. The eukaryotes, same group as before, and then the organisms that did not have their DNA packaged in a nucleus were split into two distinct lineages. One included all of the organisms that even today people would be familiar with as prokaryotes, what you would call the true bacteria, and that's what he called them. The bacteria like E. coli, or the causative agent of tuberculosis, or cholera, or anthrax, and other sort of standard bacteria that a lot of people are familiar with and study. And then this new group that was completely unrecognized previously, that Woese called the Archaebacteria. And he originally called them this because archae was meant to refer to sort of the primitive earth, and many of the organisms in this group had very unusual lifestyles. They lived in very extreme environments like boiling hot springs and acid pools and very high salt conditions. Because they are not bacteria, this group has now been renamed the Archaea, and that's what they're referred to in the book, and we will refer to them in lecture. The bacteria part of the name has now been dropped because they are no more bacteria than they are eukaryotes. But this was really significant. There was a third branch in the tree of life that nobody knew about prior to building this first tree that related all organisms on the planet. So again, what you should do is, we're going to walk through this today and in the next lecture, but you should go back and review some of the features of eukaryotic cells and some of the features of prokaryotic cells. While you do that, you should remember that prokaryotes are actually two distinct evolutionary lineages. They actually have many features, when you look at just their cell structure, in common, which is why people grouped them together previously. But as we will see, they have many features that separate them as well, and that we will see some examples of how archaea are actually, even in terms of features, very distinct from bacteria and eukaryotes. So you should start to look through these because you will have to know some of these different features. This is just an example of some of the places that they found archaea. The salt flats by the Dunbarton Bridge, for example, where they're evaporating salt. The archaea start to come in when the salt concentrations get really high and nothing else can live there. Yellowstone hot springs and these boiling acid pools. There are archaea that have now been found in more normal environments, but they do tend to dominate some of these extreme environments. So I've taken this table. You're not going to be expected to memorize this table, but this is in the book in chapter 26 in the introduction to bacteria and archaea, and it shows some of these features that are sort of conserved within bacteria, but maybe different between bacteria and archaea and eukaryotes and vice versa. You should again familiarize yourself with this table, and we're going to use some examples from that to talk about evolution of features of these organisms. In particular, look at the ones where there are some differences between the three groups, not where everybody is the same. So again, there's one curiously annoying thing about this tree, which is that there's no root. So I think I mentioned this in section before. Why isn't there a root to this tree? Anybody know? Yeah.
There's no outgroup for all life on Earth.
Right. So we have all life on Earth, the tree of life. To have an outgroup, we would have to have a lineage that separated out prior to the existence of the common ancestor of everything. if we're including all life on the planet as our in-group, we don't have an out-group. So there is no out-group for this particular tree. Or no obvious one, at least. I mean, you might have some external information that could tell you where to root this tree. There isn't any obvious external information. And that is a problem because we normally use an outgroup to root our trees. You could come up with tricks or something, but that's why Woese and other people still today draw this three-domain tree frequently as an unrooted tree. What we're going to do now for the next, say, 15 or 20 minutes is take you through a way that people have rooted this tree. A very ingenious trick. I frequently have chickened out of doing this, but it's a little complicated. It's going to take you a step back maybe before we go a step forward. But it's really important because the rooted tree of these three groups is used in the book and in many other areas as well as in class as a very important thing for understanding the evolution of features of organisms. And so the way that this tree was rooted, and you'll see this as we go through it, was a trick that relates to, in part, the ribosome. So if you take any particular organism and you start studying its mechanisms of translating proteins, so taking RNA and translating that RNA into proteins, you can find, this is a sort of diagram of a cartoon of how this is happening in a particular organism. I think this is from E. coli. There's a key function as part of this process where you're adding an amino acid to the growing protein chain. And there's a process called elongation that allows you to continue to add an amino acid to the growing chain. There are proteins in organisms called elongation factors that aid in this process. If you don't have those elongation factors, the extension of a protein building the amino acids falls apart. And so you need these to carry out protein synthesis. And when people look inside cells, for example, in E. coli, they found that there were two functionally distinct forms of elongation factor proteins. And that's what's shown here in the diagram. You don't have to understand the biochemical details. You should just appreciate that there are two different forms, but it's shown here's elongation factor 2 and here's elongation factor G, allowing this growing chain to continue to be elongated. So there are these two forms. You don't have to know the biochemistry, but one's called elongation factor G and one calls elongation factor TU. And if you ask a question, what other organisms, so this was originally found in some model organisms, what other organisms have elongation factor TU and elongation factor G? It turns out everything does. Every organism on the planet has both forms of these elongation factors. So the mechanism of translating RNA into protein, we know it's highly conserved. Ribosomes are very similar across organisms. And it turns out every organism has these two forms of this elongation factor, two different versions. So we can ask a simple question, which is, okay, if all organisms on the planet have these two elongation factor forms, what does that tell us about what was present in the most recent common ancestor of all organisms on the planet? What are we going to infer? Right, so they probably have both elongation factors. That's just the most parsimonious explanation. So if we have a tree, and here are the organisms with elongation factor 2 and elongation factor G, we're going to infer that the simplest model is that the common ancestor had both of those. And that every node along the tree, the organisms there had both of these elongation factors. It's just sort of a simple parsimony character state reconstruction. Now, the topology of this tree, just like I've shown you for the figuring out whether or not there was one or two trees of life or anything related to a universal trait, the topology of the tree does not really influence at all our inference that the common ancestor had these features. So we're not saying anything here about the order of branching among organisms. We're just saying if something is universal across all organisms, we can infer it was probably present in the common ancestor. So now we can ask a separate question, which is what the scientists who figured this out did. How did the common ancestor of everything get these two forms of elongation factor? An important part of this story is that it turns out the two different elongation factors are actually homologous to each other. So if you compare all the elongation factor 2s in different species to each other, you can line them up just like Carl Woese lined up ribosomal RNAs. All elongation factor 2s are related to each other. All elongation factor Gs are related to each other. But the cool thing is, all elongation factor TUs are related to all elongation factor Gs. They can also be lined up with each other such that they clearly are derived from a common ancestor. They're very similar in their three-dimensional structure and in their sequence of amino acids. So what people have inferred from this pattern is that sometime a long time ago, prior to the existence of the common ancestor of all organisms, there was a single elongation factor gene. And that gene was then duplicated such that an organism had two copies within its genome. And then those two copies started to become different from each other. So inside the genome of one organism, you had two different elongation factors until eventually they acquired slightly different functions. That is the elongation factor G function and the elongation factor 2 function. And if you zoom in on this tree, I'm going to show you what that duplication might look like in terms of evolution. So if we zoom in on this part of the tree, here in black, I have drawn the species evolution. So here in some lineage, in the root of the tree, so in some common ancestor of everything, although not the most recent common ancestor, which is here, there was one elongation factor gene. That gene then duplicated into two genes. And those genes eventually accumulated differences from each other. So you can actually have two different forms of a gene within one genome becoming distinct from each other. Eventually, the common ancestor of everything they became these distinct functions, EFTU and EFG. And then after that, there were speciation events in the lineages leading up to bacteria, archaea, and eukaryotes. Every descendant of that point inherited both forms of the gene. So you can draw that like this. I've drawn, in essence, an evolutionary tree of genes inside this evolutionary tree of species. So the blue represents the EFTUs, and the green represents the EFGs. This can be extended all the way to modern times. That's how a model for how all organisms got two forms of this gene, and the genes are related to each other. There was an ancient duplication event in the history of this gene family. Are there any questions about that before we go on to how we use this to root the tree? Okay, so the cool thing about this is we can now analyze the evolution of these genes to learn about the evolution of species in which the genes are present. And that's what I'm going to show you here. So if we take that tree that I had drawn, we can extract these trees of the individual genes from that species tree and look at what the trees of these genes should look like. So if I throw away the species tree and I unwrap the gene tree by pulling the elongation factor 2s and elongation factor Gs into their own corner here, that is what the evolution really looks like. We get a tree that looks like this. All EFTUs are related to each other and share a common ancestor with each other. All EFGs are related to each other and share a common ancestor with each other. And the point at which EFGs and EFTUs share a common ancestor together is that gene duplication event, that point. So now there's a really cool aspect of gene evolution that we can take advantage of, which is we now have outgroups. So if we look at the EFGs, What sequences, what genes in here are outgroups to the EFGs? Is there anything in our tree that branched off in the tree prior to the existence of the common ancestor of all the EFGs? Yeah, so all of the EF2s, here's the common ancestor of the EFGs. What we need to look for is a branch in the tree that is below that branching point. Here it is. It's the branch leading up to all EF2s. So all EF2s can be considered an outgroup for if we're studying the EFGs. So if our in-group are the EFGs, the outgroup relative to that, it could be any and all of the EF2s. The inverse is true for the EF2s. Any and all of the EFGs are outgroups for the EF2s. So where is the root on this tree? Anyone know where the root of this gene tree is, as I've drawn it here? Yeah. So this is the root. Is that what you're... So this is the root of a tree. So it's really important that you realize that a phylogenetic tree can be drawn for species, which is everything we've been talking about prior to here. We can also draw a phylogenetic tree like this for genes. It also reflects history. In this case, it's the history of these genes, not specifically only the history of species. But we've drawn it as a horizontal tree. For this horizontal tree, this is the root branch of that horizontal tree. So here's the common ancestor of everything in our tree. That is all the EF2s and EFGs. And here's the branch leading up to that common ancestor. If we go back and look at the species tree, this root branch is, in essence, the branch leading up to the duplication event that occurred in a common ancestor of all these organisms. I'm going to go back to the gene tree here. So here's the root of that gene tree. So with that in mind, I know this is complicated, but with that in mind, we're going to take one more step. So the scientists that were interested in that said, okay, that's a good model. We like this model where there are two different types of elongation factors, EFG and EF2, and we think that they're related to each other by this duplication event that occurred prior to the existence of the common ancestor of everything. So let's build a tree of all elongation factors, where in one tree we're going to include elongation factor 2s and elongation factor Gs. So this was a hypothetical tree. That is, if we have this, I had this model that I was explaining about duplication, let's see what real data actually looks like. So they took sequences of elongation factors from different taxa, aligned them to each other, and built an evolutionary tree. Here is a sequence alignment taken from one of these papers, actually. And here's what the tree looked like. And I've labeled B refers to bacteria. The number refers to the number for a particular taxa. So B1 is a gene from species bacteria number 1. There are 2 B1s on this tree. You'll see what that means in a minute. There are 2 B2s. I included 3 Archaea here and 3 Eukaryotes. On this part of the tree are all of the things that function as elongation factor 2s. I already told you they're all more similar to each other than they are to anything else, so this is good that in our evolutionary tree we can draw, a grouping around the EF2s. Here are the EFGs. Each species has one of each. So eukaryote number 1 has an EF2 and an EFG. Eukaryote number 2 has the same. Number 3, Archaea number 1, number 2, number 3, bacteria number 1, number 2, number 3. So each species has one of each. So now we have this tree, an unrooted tree, showing the relationships among elongation factors 2 and elongation factor G from these different species. How do we root this tree? Where should the root go on this tree? Yeah?
Should it be in the middle of where the EFG and the EF2s are?
Right, so it should be in the middle here because if our model of the duplication is correct that I drew, that root branch was in the branch leading up to the branch that connected EF2Us and EFGs to each other. So the root should go here. If we now place the root in the tree there, and we unwind this unrooted tree just like we unwound the unrooted trees with 4 taxa, I realize this is more complicated. If we unwind that, we get this tree. Here's the root that we drew. That node is at the gene duplication event. Here are the EFGs and the EF2s. Here is showing them side by side in case you want to compare to make sure that I didn't invent stuff here. So there's node number 1. We can show node number 1, node number 2, etc. all throughout the tree. Everything lines up. So remember, the EF2s, this is what the rooted tree would look like for these genes. We can also just look at whether or not the EF2s are outgrouped to the EFGs and vice versa. They are in this tree. Then what we can do is look at the relationships within the EF2 branch. So how do the taxa relate to each other? I didn't just invent this. How do these taxa actually relate to each other? And what you see is there's a branch leading up to the common ancestor of the 3 bacteria. And Archaea and Eukaryotes share a common ancestor here. Or if we collapse all the lineages, this is what the tree of EF2 looks like when we've rooted it in between EF2 and EFG. And if we look at the other side of the tree, it looks the same. So what this means is if our model is correct for this ancient duplication event, when we root the tree in between EF2 and EFG, we get bacteria as the deepest branching lineage, and Archaea and Eukaryotes as sister taxa. This type of analysis is why, in my lectures and in the book and in many other places, some people consider Archaea to be the sister taxa to Eukaryotes, to the exclusion of bacteria. This analysis is not very statistically significant. There is a lot of noise here when you're studying events that happened 2 or 3 or 4 billion years ago. There's a lot of strangeness that happens. There is some suggestion that this might be the true relationships among organisms, but it is a very weak suggestion. So many people still draw the tree of life as an unrooted tree because it's unclear if this pattern is correct or not. And it's still an active area of research. So if we take that pattern, we can redraw the tree. Oh, here's the actual tree from one of these original papers. Turns out people did this with other forms of duplicated genes and got the exact same result. It's still a little bit ambiguous, so people are still not completely confident in it. But different types of duplicated genes showed the same pattern. So in essence, we can draw the tree of elongation factors like this. Eukaryotes and Archaea together with bacteria as the deepest branch. And here's that duplication event. I'm going to skip over this definition. You don't need to worry about it. So for the next few minutes and all of the next lecture, when we talk about this in more detail, this is the tree we're going to use. And that duplication is why we did it that way. People analyzing duplicated genes like that suggested that this is the rooted tree of life. That is why we're going to use it for analyzing the patterns of features in these different organisms. So are there questions about that rooting the tree of life before I move on now to using this rooted tree of life to study features of organisms? You know it's a little tricky, but you have to think of individual organisms have thousands of genes. You can imagine inside the evolution of species, the genes are evolving in their own particular way. And occasionally you get this weird thing where 2 genes emerge from 1. And then you can study the evolution of each of those lineages within individual species. Okay, so what we're going to do now is switch to using this rooted tree of life to study and learn about the features of these three lineages. So again, you should look through that table in the book and those pages of the book from chapter 1 and chapter 4, etc., to familiarize yourself with some of the key features of eukaryotes versus prokaryotes, and then the differences between some of the different branches of each of these organisms, like archaea versus bacteria. So just as a little aside before we get into this, I've already heard some questions about this. Some people call bacteria primitive or archaea primitive. We don't actually know, in many cases, what any of the ancient organisms looked like. There is some fossil data, so we might be able to infer what ancestral character states were like. And we could use this terminology, which I don't really like, to refer to, say, bacteria having an ancestral feature and calling them primitive. What I'm going to show you for the next few minutes is some of the ambiguities as well as some of the things we can learn about what the ancestral character states might have been in organisms without using the fossil record, just by analyzing the tree and data from organisms. So here's another table with some of these features of bacteria and archaea versus eukaryotes. For this table, I've shown bacteria and archaea being identical in everything. They are actually different in some features, but overall they have many outward similarities with each other, which is why people group them together for a long time. And whenever I talk about things, they're probably every single one of them there are going to be exceptions to them. There's sort of generalities, that is 99% of bacteria might have one feature, probably not all of them. So I'm going to just skip over this because I'm going to go through the individual examples of each of these. So again, look at the cell of a eukaryote and the cell of a prokaryote. There are many differences. And what we're going to do is study the evolution of some of those differences. So we're going to take this rooted tree, and we're now going to pick some features that are worth mapping onto this tree. Remember when we show this rooted tree that each of them implies sort of a large collection of other organisms? So I'm not going to show all the different branches of bacteria when I show this, but this tree implies that there are lots of other lineages of bacteria, archaea, and eukaryotes there. Sometimes I will show this sort of tree with lots of different things. So let's first consider features that are unique to one of these three lineages and widespread in that lineage. That is, basically present in all the members of that lineage, but different in other taxa. And another really important thing, because I've already gotten this question a few times, if we look at any of these features, let's just say the presence of a nucleus, we are not using these features to infer the relationships among these organisms. We are not using these features to figure out the rooting of the tree. We are taking the tree from the ribosomal RNA tree and from that rooted tree that I showed you, and now tracing the evolution of features on a particular tree. Because you will see conflict among many of these characters and the tree that we drew. And that conflict will most likely be due to homoplasy. It doesn't mean that the tree is wrong. We're using that tree, and then the characters might evolve multiple times, for example. So let's take one that you may have heard of, peptidoglycan. Peptidoglycan is a component of the cell wall of bacteria. It is actually the target of some antibiotics that target bacteria. It's a long polymer, basically, of this sort of complex between amino acids and sugars. And archaea also have a cell wall that is so bacteria have a rigid cell wall, most of them. There are a few that don't. Archaea have a rigid cell wall, most of them. There are some that don't. The bacteria's cell wall is made up of this peptidoglycan compound. Archaea use a different compound called pseudopeptidoglycan. Seems to be of a distinct origin, maybe completely separate origin. So let's map this feature onto the tree. All bacteria have peptidoglycan. So I've drawn it as present. Archaea and eukaryotes do not. So where do we think the peptidoglycan feature, how can we infer the ancestral character states for peptidoglycan? One possibility is shown here. It was present in the common ancestor of everything, and then lost on the branch leading up to the common ancestor of Archaea and eukaryotes. That requires one change event. An alternative possibility is it was absent from the common ancestor of everything and gained on the branch leading up to bacteria. That also requires one change event. Which do we pick? Does anybody want to say which one they pick?
Isn't there a third choice?
What is the third choice?
Well, wouldn't it be that it could have developed after the ancestor to Archaea, or that it could have changed? Like it could have evolved separately between Archaea and Eukaryotes, which is why Archaea group would have had like a pseudo version.
Yeah, ignore the pseudo, just pretend like it's completely different. It's a misterminology. It really does seem to be a separate origin.
Okay.
So you could actually have had a loss in the archaeal branch and a loss in the eukaryotic branch. That's going to require two changes, so we're going to assume that that's not as parsimonious as this, which requires one change. But you can't really easily distinguish between these two alternative theories given the model that I've just talked about here. So we don't have a really good model to say which one of these occurred. There are some arguments you could make for maybe this being less probable. That is, once it was gained, maybe it would be unlikely to have been lost. But really, given this tree and this feature, and in fact, any features that are unique to and widespread in bacteria and absent from Archaea and eukaryotes, it's going to be hard to figure out what the ancestral character states were, given this tree. So the tree actually tells us what we don't know here. So let's look at unique archaeal features. One interesting unique archaeal feature is the exact chemical structure of the membranes. So all cells are surrounded by a membrane structure. The membrane in all cells has a what's called hydrophobic part. That is the part of the membrane that avoids water. And a hydrophilic part. That is the part of the membrane that dissolves in water, in essence. And the structure, the chemical structure of the membranes in bacteria and eukaryotes is highly similar to each other. I have them down here. So here's bacterial and eukaryotic lipids up here that use, don't worry about the chemical details, they use something called an ester bond in their membranes. And Archaea use a distinct chemical bond and actually synthesize their membranes in a very distinct pathway, something called ether linkages in their membranes. It's considered an indicator of Archaea in many cases. There are exceptions. Some bacteria use ether linkages, but most don't. If you find fossils, for example, and you want to figure out what type of organism they are from, you can actually look for signatures of these membranes to try and tell you if something was an Archaea or a bacteria or a eukaryote. It is considered a sort of hallmark feature of Archaea. So let's map that onto the tree. Here's a feature that Archaea have that's different in bacteria and eukaryotes. What are the different evolutionary scenarios? It could have been ancestral. Given the tree we have here, that would require a separate loss event or change event in bacteria and eukaryotes. It could have been gained in the common ancestor of Archaea and eukaryotes and then lost in eukaryotes. Or it could have been gained in the lineage leading up to the common ancestor of Archaea. This one only requires one change event. So, it's 1 versus 2, but this is a more parsimonious explanation. So features that are unique to and widespread in Archaea, we would conclude most likely originated in the branch leading up to the common ancestor of all Archaea, after it separated from the lineage that it shares with eukaryotes. So there turns out there are some features that are shared between archaea and eukaryotes. One of them is histones. So in eukaryotes, you may be familiar with this, the DNA is packaged into chromosomes. The DNA is actually wrapped up into these bundles, and the bundles are wrapped up into coils of bundles. The wrapping is done by wrapping the DNA around proteins called histones. And even though people thought that histones were a hallmark feature of eukaryotes, they have been found in archaea. And as far as I know, they've never been found in bacteria, although there are things that occasionally resemble histones but are probably not homologous in some bacteria. So for this feature, present in archaea and eukaryotes and different in bacteria, we have a few scenarios. It was ancestral and changed in the bacterial lineage, so maybe all organisms had the common ancestor had histones and then bacteria invented something else and got rid of them. Or it was invented in the lineage leading up to the common ancestor of archaea and eukaryotes. Which one wins? They're equal. So we can't really distinguish with this tree for those features. So the last thing I want to talk about today is probably the most interesting to many of the people here and will be something that you will hear lots about, both in the course and in other places, which is the unique eukaryotic features. For example, the nucleus. So the packaging of DNA into this membrane-bound compartment, this nucleus, is a unique feature of eukaryotes, not present in bacteria and archaea. How would we map that on the tree? Here's a unique feature of eukaryotes. There are many different possible explanations for its evolutionary history. The most parsimonious is probably going to be the one where it originated in the lineage leading up to the common ancestor of eukaryotes. And the thing you should think about when you read those parts in the text is there are an enormous number of features like this that are unique to eukaryotes and distinct from bacteria and archaea. People tried to build evolutionary trees from these features, which is why for many years they didn't recognize the existence of the archaea. But most likely these features evolved in the branch after the common ancestor of archaea and eukaryotes separated, prior to the existence of the common ancestor of all eukaryotes. So these features do not tell us that archaea and eukaryotes are closely related. They just tell us that eukaryotes have invented all sorts of things since they separated out from the common ancestor they shared with archaea. And we will talk about in the next few lectures in more detail many of these features. Oh, we just switched it over to 2.